Assalamu alaikum. My name is Faryal Razak, uh, and my complete roll number is 1EP1770038. My group number is 5, and today the topic of my presentation is basically the exploratory behaviors of the rodents. If we talk about the exploratory behavior, first we will talk about what is a behavior. We can define the behavior is basically any evolved adaptive action or an activity of an organism that interacts with its environment, not simply a byproduct or side effects of an activity. Behavior can be learned or inherited. Basically, we have two types of behavior. First one is the innate, and the second one we can talk about is basically the learned behavior. If we talk about the innate behavior, that is, it means basically the inherited instinctive, it is also an automatic and the consistent learning behaviors. If we talk about the learned behavior, it is basically the ability to learn is inherited, but the behavior develops during the animal's lifetime. It is variable and the flexible, that will change with the time or experience and the environment as well. Now, what is the animal behavior? It basically includes all the ways animal interacts with the other organisms and the physical environment, and we can also say with the drugs as well. Different behaviors shown by the animals. Different animal shows the different type of behaviors. Some of them are basically the learning behaviors. Some can show the reproductive behaviors, the locomotion behaviors, animal communication, aggressive behaviors, feeding behaviors, and the avoidance behaviors as well, etc. So what is the ethology? The study of individual behavior patterns is known as the ethology. Now we have models based on the exploratory behavior. We have very different types of the models, but some of them is are basically that are used for the exploratory behaviors for the rodents are the light and dark box, zero maze, open field, elevated plus maze, whole boat, staircase, free exploration tests, and many more. But today the topic of my presentation, or the focus of my presentation, is basically the light and dark box technique for evaluation or the determination of the exploratory behaviors of the rodents. So animals basically placed in the two chambered system where they can freely move between a bright and the dark compartments. The number of crossings between the light and the dark sites is recorded as well. If we talk about the apparatus of the light and dark exploration model, a dark and a light chamber divided by a photosil equipped zone. This case rests on an activity monitor which counts total locomotory activities, and the cage is about 21 into 42 into 25 centimeter. The light intensity is different in the two chambers. If you talk about the light intensity in the light chamber, that is about 390 lux, and if you talk about the light intensity in the dark chamber, that is about 2 lux. Now, what are the requirements for our technique, basically, which we are using for the exploratory behavior, is basically that the male mice with a weight of between the 18 and 24 grams are used in the experiment. Test drug administered 30 minutes prior to the experimentation by the intraperitoneal route. The animal used in the experiment is placed in the cage. Now, what is the method of the light and dark box by which we are performing our experiment and evaluate the different behaviors of our rodents? First, the subject is placed in the dark portion of the box for the set period of acclimation time. At the time of this period, a door separating the two compartments is being opened. The door is used so that the mice do not enter the light chamber immediately after the release with their motivation to escape from experimenter, since the latency to enter the light chamber may serve as an index of anxiety-like behavior. The amount of time that the subject takes to emerge fully from the enclosed area into the open area, and the percentage of uh, time spent in the dark and the light chambers are being measured as well. The distance traveled in each chamber, the total number of transitions, the time spent in the each chamber, and the latency to enter the light chambers are being measured. Now, if you talk about the drugs that is used in our experiment of by which we can explorate the behaviors of the rodents, are of basically many classes being used, but the main class, or the class which is being discussed by myself, is the benzodiazepine class drugs are commonly used. They are used to relieve the anxiety like behaviors in the rodents. If you talk about the site of action, it basically affects the midbrain, ascending reticular formation, and the limbic system. And if you talk about the mechanism of actions of the benzodiazepines, they may mainly affect or work by post-synaptic inhibition through the benzodiazepine receptors. And if you talk about, we have very different drugs included in this in the class of the benzodiazepine, but some of them are the diazepam, oxazepam, lorazepam, etc. If you talk about the lorazepam, it is basically given orally and IM. It has no active metabolites. It has a very short acting, and the preferred in the elderly patients mainly, used in the short lasting anxiety, panic, OCDs, tension syndromes, and the dose is between 1 to 6 milligram per day. We can just administer the dose between the 1 to 6 milligram per day. If you talk about the oxazepam, the hepatic metabolism is less significant. It is preferred in the elderly and those with the liver diseases. It has a short duration of action, and it is used in the short lasting anxiety states. Now, I'm just gonna discuss the another technique that is whole board method, which is also one of the standard methods. And the purpose of the whole board method is the evaluation of the certain components of the behaviors of mice, such as the curiosity or the exploration. They used in the open field with the holes on the bottom into the which the animals could poke their noses. um, Now, what is the procedure by which we are just gonna perform this experiment, that is mice weigh between the 18 to 22 kilograms. The whole board has a size of about 14 to 40 centimeters. 16 holes with a diameter of about 3 centimeter each are distributed evenly on the floor. The board is elevated as so that the mouse poking its nose into the hole. The nose poking is thought to indicate the curiosity, and the motility measured in the addition by counting the interruption of the light beams. Now, what the comparison, we are just gonna compare my method. I'm just gonna compare my method exactly with that of the standard method, or with one of the standard method, that is the whole board. So the whole board is a standard method as compared to the light and dark box. The whole board gives about 58% results, and the exact percentage of the light and dark box is unknown yet. There is a huge difference between the apparatuses of the both methods, and the results are also very different from these two techniques as well. Now, the last and the very important thing we want to discuss, or I'm just want to discuss over there, is the article related to the light and dark box. The article which I have used for my presentation has an abstract, that is the light and the dark transition test, which is also known as the LDT, is one of the most widely used tests to measure the anxiety like behaviors in the mice. The test is based on the natural aversion of mice to brightly illuminated areas, and on their spontaneous exploratory behaviors in the response to mild stressors such as the novel environment and the light. This test is also sensitive to anxiolytic drugs treatment. The test apparatus consists of a box divided into a small one-third dark chamber and a large two-thirds brightly illuminated chambers. Mice are also placed into the light compartment or the chamber and allowed to move freely between the two chambers. The first latency to enter the dark compartment and the total time spent in the light compartment or chamber are indices for the bright space anxiety in the mice. Transitions are index of the activity exploration because of the habituation over time. LDT is very quick and easy to use without regarding prior training of the animals. Here we have no requirement to train our rodents or our mice. So we present our protocol that has been able to detect both anxiolytic like, or we can also say that reduce the anxiety like behaviors, and anxiogenic like behaviors in the mice, that is increased anxiety like behaviors. Now, what are the materials and the reagents which are we just gonna use in the experiment? That is the paper towels, laboratory bred mice, and then they are must be in the groups of the 3 to 5 per cage, kept in the room with the controlled temperature of about 23 degrees centigrade, and the humidity under the 12 or light to dark cycle, and with aid libitum access to the food and water. And the third thing which is important, that is the 70% ethanol. Now, if you if you want to talk about the equipment that is used, or the details of the equipments that is being used in the experiment, is the apparatus for the light and dark transition test consists of a box of about 42 into 21 into 25 centimeters, divided into a small one-third dark compartment and the two large, that is two-third illuminated compartment. Secondly, a restricted opening 3 centimeter high by 4 centimeter wide connects the two chambers as well. If you talk about, as we can see the figure 1 that is right in front of you, it has basically the two chambers, and there is a camera that is placed exactly upon over the apparatus which records the movements of the rodents or the mice. So if we talk about the figure 1, there is an indirect white light source, digital lux meter, video camera that is placed directly above the apparatus, digital chronometer for the manual analysis, or we can also use the computer for the automated analysis as well. If we talk about the procedure, the apparatus is placed in an isolated room away from an external interferences and the noise with a low intensity white light source. The experimenter must restrict from making any excessive noises during the test and from wearing odor and products with the very strong smells as well, since it could act as the anxiogenic stimulus for the mice or the any rodents. The illumination in the light chamber must be between the 200 to 400 lux or more, while the other chamber is dark and having a 5 lux or less intensities. The mice are transferred in their home cages to the behavioral testing room for at least about 3 minutes before the experiment. It is very a basic need of the experiment. Now clean both the compartments of the apparatus with the 75% ethanol, turn on the camera and place the first mouse in the middle of the brightly illuminated chamber. The experimenter stays as far as possible from the box out of the side of the test animal. The mouse is allowed to move freely between the two chambers for 5 minutes. After each trial, all the urine and the fecal villi are removed from the both the chambers are cleaned with 70% ethanol. The recorded videos can be analyzed by automated computer software or manually with the aid of the chronometer. The following parameters are scored as shown in the figure 2. There is a representative data or a figure 2 which is right in front of you. I'm just gonna say we have 4 sets of different data that we have collected from this experiment. Each set of data represent the different results, and if you talk about the data A or the set A, that shows the latency time to enter the dark compartment. The set B or the data B, this basically describes the time spent in the light or the dark compartment. The data C or the chamber C, which basically describes the distance traveled in the light chamber, and the D, that is the number of the transitions as well. Now there is a conclusion, or we can say that the very important points that we have to keep in our mind are the are I'm just going to discuss in a very brief manner. That is, the LDT was originally developed by the Crowley and the colleagues, that is the Crowley and the Goodwin, in the 1980. There are two differences between the original version and our test. The first one, that is the duration of the test is about 30 minutes, 10 minutes in the original version, whereas we used only 5-minute testing time in our this type of experiments. And secondly, the difference is that in the original version is that the walls of the lid chamber are transparent, while we use the opaque white plastic for the walls. Thus our test protocol is more specific for detecting the bright space anxiety, and compared to the original version where also open space anxiety is scored as well. Secondly, the typically male mice are used for the behavior experiment. Now, what is the reason of using the male mice is basically as variation of the estrous cycle might influence the performance of the females in such tests. Baseline activity we may vary according to the mice strain or the mouse strain and the age as well. The fourth one, that is in order to ensure uniform luminosity, and indirect light is used to avoid the production of the shadows, which can be a place of preference for the mouse. Fifth one, that is the both compartments must be always cleaned after the testing of each animal and fully dried after cleaning, because the strong smell of the cleaning agent might alter the mouse behavior as well. Uh, the sixth and the last one, that is an entry into the light or the dark compartment, is only considered when the animal placed between the two frontal paws inside it. If the animal returned with these two paws into the compartment, no entry is being registered. Now, what are the acknowledgements about of the experiment? That are the protocol described here has been adapted from the previous study, which succeeded in the identifying anxiolytic like behaviors for the mice. This work was basically supported by the grants from the German research and the KB, the European Union FP7 program, and the German Ministry of the Research and Education. So now at last we have different references by which I have collected all the data for the presentation. So in last, I would like to end my presentation or summarize my presentation, or just that note that we have very different methods or very different techniques which we can use basically to check the exploratory behaviors of the rodents, but some of them are the standard methods, and some are not included in the standard methods. But so that's why basically there is a difference between the results percentage.